This is Micro Unit 7, Part D of the lecture. Acquired immunity develops with exposure to an antigen. It is naturally acquired active immunity, and the body responds to the antigen or pathogen with a specific immune response to exposure from the environment. Naturally acquired passive immunity occurs with the IgG that crosses the placenta and the IgA from breast milk, providing early protection to infants. The immunity is passive because the infant is not producing its own antibody. In artificially acquired immunity, the immunity is induced by introducing an antigen in the form of a vaccine, and the patient's immune system actively responds. Artificially acquired passive immunity is used when the active immune response is too slow, such as to snake venom. Antibodies are harvested from the blood of immune humans or animals and injected as antitoxin or antiserum. Active immunizations are administered to stimulate an immune response. Passive immunizations provide temporary immunity through the transfer of antibody from immune individuals. The shortest term protection is going to come from that artificially acquired passive immunity, although it is able to save a person's life in many cases, such as from snake venom. Historically, it was noted that children who recover from smallpox did not get it a second time. Early vaccinations involved deliberately infecting young children with ground-up smallpox scabs. It's not a real appealing idea, but grinding up the scabs was a good way to get them exposure to the disease. The reduced mortality and morbidity was found with the children who consumed these scabs. Edward Jenner demonstrated protection from smallpox with inoculation with crusts with an infected person injected with cowpox, which was antigenically very similar. Louis Pasteur conducted experiments with the Pasteurella and demonstrated effective vaccines against the organism. By the late 1900s, we had formulated vaccines for polio, measles, mumps, and rubella in the United States. We now have vaccines for chickenpox, hepatitis A, hepatitis B, and others. Kind of a scary idea, but a lot of the early microbiologists, especially in immunization, actually tested their first patients were their own children. Not all types of vaccines are equally safe or effective. Live attenuated vaccines reduce the virulence of the organism so it is still living, but no longer causes disease. They are grown in tissue cultures until they lose their virulence or are genetically manipulated. Live attenuated vaccines still pose a risk in pregnant women or immunosuppressed. Inactivated vaccines use formaldehyde to inactivate the agent. Whole agent vaccines use deactivated whole microbes. Subunit vaccines use antigenic fragments of microbes. These are safer because there is no organism to replicate, but boosters are needed. Adjuvants are substances that increase the effective antigenicity of a vaccine. They may also stimulate inflammation and allergy. Killed vaccines are recognized as exogenous antigens and stimulate a humoral response. So as you know from your training in our class, formaldehyde is a carcinogen or a potential carcinogen, strong potential carcinogen. And so there is some toxic component to the vaccines, but formaldehyde is the one way we do have to kill the organisms and to preserve them, which is needed to stimulate the immune system safely. Toxoid vaccines are chemically or thermally modified toxins in vaccines to stimulate immunity. Both the tetanus and diphtheria vaccines stimulate antibody-mediated immunity. They have few antigenic determinants, so they need to have multiple doses and boosters using recombinant DNA technology. So when you get a tetanus or the diphtheria vaccine, what you're actually getting is a vaccine to the toxin that is produced, not to the organism itself. We can detect the virulence genes and can produce pure bacterial and viral antigens, but they are less effective. Genetically engineered microbes can be used as vehicles. The CDC has a recommended schedule of vaccinations, which is regularly updated with new and changed vaccines. With active immunizations, as with any procedure or drug, there are some risks. There may be symptoms associated with the mild toxicity of the vaccine, such as pain at the injection site, malaise, or fever. A more severe allergy to a component of the vaccine may result in anaphylactic shock. In the case of live vaccines, the residual virulence may cause disease. These are reported and recorded through the Vaccine Adverse Event Reporting System. At this point with polio, almost all existing cases of polio out there are actually due to the vaccine. Antiserum contains antibody for passive immunization. This is typically collected from large animals intentionally exposed to the disease. There are some limitations to the use of antisera as well. Antisera preparations contain antibody to many antigens, not just the antigen of interest. Repeated injections may also trigger an allergic response. Preparations may contain viral pathogens. The protection is short-term since antibody degrades quickly with a half-life of about 3 weeks. So a person would not want to receive unnecessary passive immunizations just because of the risk of developing an allergy to it when they really need it later on. Hybridomas and monoclonal antibody is sometimes used to make antiserum. The hybridomas are tumor cells fused with antibody-secreting plasma cells to produce clones that create a large amount of a single antibody. Monoclonal antibody is identical to antibody secreted by clones originating from a single plasma cell. Immunotesting uses a known antibody to detect an antigen, or uses a select antigen to detect the presence of an antibody in a patient's blood. Serology is the study and diagnostic use of antigen-antibody interactions in the blood serum. Precipitation tests use antigen-antibody mixed together. When they are complementary, they form a lattice-like precipitate. In immunodiffusion, or the Australoni test, 2 wells are made in an agar gel. The wells are filled with antigen and antibody and allowed to diffuse. When they meet, if a precipitate forms, they are complementary. A similar type of reaction can be observed on a larger scale in immunoelectrophoresis. The antigen is separated by electrophoresis on a gel, and a trough is cut parallel to the antigen in the agar. Antiserum is placed in the trough and diffuses laterally, and a curved line of precipitate is formed. This technique can resolve more than 30 distinct antigens at once. Agglutination tests still take advantage of the antibody cross-linking a particular antigen, but involves clumping the soluble molecules so they are easy to see and interpret without visual assistance. It can be done with serial dilutions to determine the titer of antibody with agglutination activity. Neutralization tests are based on the ability of an antibody to neutralize the biological activity of many pathogens and their toxins. This can be done with neutralization or viral hemagglutination inhibition tests. The virus is mixed with specific antibodies, and it lacks their specific cytopathic effects when introduced into a cell culture. Complement fixation tests are used to detect the presence of antibody in the serum too low to be detected with agglutination. The serum sample is heated to 56 degrees Celsius to destroy naturally present complement. Antigen from the disease in a standard amount of complement is added. Sheep red blood cells and antibody against them are added. In the presence of antibody against the antibody-antigen complexes bind complement, which then cannot bind anti-red blood cell antibody to lyse the red blood cells. In the absence of antibody, it leaves the complement free to bind the anti-red blood cell antibody attached to the red blood cell, and cells are lysed, making a clear mix. A positive test is cloudy. Labeled antibody tests use antibody linked to a molecular label. This allows them to be easily detected. In direct fluorescent antibody tests, the tissue is flooded with radio-labeled antibody, and the tests are used to identify the presence of an antigen in the cell tissue or serum. The antigen is fixed to a slide, and the patient's serum added, and then radio-label antibody is added. ELISAs are enzyme-linked immunosorbent assays. In this test, the label is not a dye, but an enzyme that reacts with a substrate to produce a color product, indicating a positive test. Wells in a plate are coated with antigen. The excess antigen is washed off, and another protein, such as gelatin, is added to coat the polystyrene surface not coated with antigen. The antiserum is added. If the antibody is present, it will bind the antigen. The anti-antibody labeled with an enzyme is added. The enzyme-substrate reaction produces a visible color change. Western blots are used to detect antibody against multiple antigens in complex mixtures. They are used to verify HIV tests and ELISAs. It's very common to see the HIV or the ELISA and Western blot tests used together because it provides more accuracy overall. It detects more types of antibodies and has less misinterpretation than the ELISA. It starts with antigen in solution being separated by gel electrophoresis. The protein band is blotted onto a nitrocellulose membrane and then cut into strips. They are washed and exposed to the serum, followed by being washed again, and then exposed to antibody. Again, only the antibody sticks to the antigen and the corresponding anti-antibody. It is washed again and exposed to the substrate and observed for color change. Immunofiltration assays are rapid ELISAs. They use antibody bound to a membrane filter instead of a polystyrene plate and the larger surface area to reduce the time required. Immunochromatographic assays use antigen solutions such as the infected blood flowing through a porous strip. As the antigen flows through, it encounters antibody labeled with pink colloidal, gold, or blue colloidal selenium. Here, the antigen-antibody complex binds to a colored immune complex and form a pink or blue line. This is what is used for pregnancy tests to detect levels of HCG.